Let's talk about muscle tissue. First, we're going to look at the connective tissue of muscle. We'll start on the outside. So the epimysium is going to cover the entire muscle. So if we look over here, we can find that. So there's epimysium around the whole muscle. Then we have the perimysium, which is going to go around a fascicle or a bundle of fibers. And then we have, if you look even further closer, we have the endomysium, which is going to be around a single muscle fiber. So again, so there is a pattern here. So epi, peri, endo, going from the outside in, just like we have the same pattern with the connective tissue around nervous tissue. We also have fascia here, so you can see right up here. So this is connective tissue that separates neighboring muscles or muscle groups from each other, and the subcutaneous tissue. Here you can see the perimysium that's wrapping around the fascicles, and here you can also see the endomysium, which is wrapping around each fiber. Here we can see the structure of a skeletal muscle fiber. So this is a muscle cell or a muscle fiber here. You'll notice that on the outside we have it's called the sarcolemma, which is the plasma membrane of a muscle fiber right there. Then we have inside we have the sarcoplasm, which is the cytoplasm of the muscle fiber. We also have these myofibrils right here. So these long cylindrical things are the myofibrils. So all of this is a myofibril here. These are long protein cords that are occupying most of the sarcoplasm, and you see we have quite a few of those. So skeletal muscles are also very multinucleated. Here we can see a nucleus there, and there. We also have many mitochondria right here. We need a lot of ATP for muscle contraction. So one of the two big things we need is ATP and calcium. So you can see the mitochondria, very necessary for muscle contraction because we need a lot of that ATP. So why do we have so many nucleuses, nuclei? It has to do with development. So myoblast stem cells that fuse to form each muscle fiber early in development. So these are actually different cells fusing to form the cell here. So here's a few more structures that we need to discuss. So the sarcoplasmic reticulum is a smooth endoplasmic reticulum, but this is for a muscle cell. This SR, sarcoplasmic reticulum, you can see it right here. So these blue things here, this is the sarcoplasmic reticulum. And then notice at the end we have these dilated parts of it. This is going to be known as the terminal cisternae, which is going to act as a calcium reservoir. It's going to release calcium through channels to activate contraction. So remember I said that ATP and calcium are really important for muscle contraction. Here's where we're going to concentrate our calcium, and then we're going to release it from these terminal cisternae. Also we have T-tubules, which are tubular infoldings. So right here, if you look closely, so this purple is a T-tubule, transverse tubule, and actually there are going to be these tunnels that go down from the sarcolemma, from that plasma membrane of the muscle cell. It's going to go down. There's holes that go down into T-tubules, which is going to help with muscle contraction. The action potential is going to travel down into here, and it's going to cause actually the terminal cisternae to release that calcium into the cell. And a triad is one T-tubule and two terminal cisternae. So right here there's a terminal cisternae around this side, terminal cisternae here, and then in the middle we have a T-tubule. So now let's talk about myofilaments. So we have myofilaments. Specifically we're going to talk about thick filaments first. So this is the thick filament here, which is made up of the protein myosin, and this is the thin filament here in red that is made up of the protein actin here. So specifically right now we're talking about thick filaments, so we're going to talk about myosin and those myosin heads. So you can look at here, we have those myosin heads. Those are going to come up and they're going to actually bind with the actin protein here, and it's going to cause movement. Actually the sarcomere is going to contract. That's what we're going to look at a little bit later here. So each molecule is shaped like a golf club, so definitely that looks exactly like a golf club up there. So I always think of Myosin heads, they have, so myosin has those myosin heads, so that has to be the thick filaments, and these big golf club heads sticking up, so that's got to be the thick filament. That's how I keep that straight. Now we have the thin filaments. So the thin filaments are made up of the protein actin. We also have a few other things going on here. We have troponin and tropomyosin, so very important with muscle contraction. We have troponin here. Calcium is going to bind with troponin, and it's going to move tropomyosin out of the way, because underneath tropomyosin here we have some active binding sites that the myosin heads, those golf clubs, can come up and bind here, and they can do a power stroke, just like if you were grabbing a rope with one hand tied behind your back and you're pulling that rope towards you, you would grab, pull, and then release, grab, pull, release. Those myosin heads are going to be doing a very similar action here. So there are binding sites that you can't see here, but when calcium binds with troponin, then it's going to move tropomyosin out of the way, and that'll expose those active sites. So the sarcomere is the contractile unit of a muscle. So right here, sarcomere is from Z disc to Z disc here. So that is a sarcomere. We are going to see muscle striations because of the alternating dark A bands and the light I bands. So if you take a look here, so an A band is going to have the myosin with those myosin heads in there, that myofilament, and the I band will not. So here's the I band right here, and you can keep this straight because alternating dark has A in it, so those are A bands, so the dark A bands, and then light has an I in it, so I bands. So you can keep that all together. So here's an animation up here of how the sarcomere contracts. So in the middle we have the myosin. We have all those little heads that are going to be pulling on that actin to contract, to bring those Z lines or Z discs closer together. So here we can look at it underneath the microscope here. So you can see all these features under the microscope here. This is a high-powered microscope, but you can see these features. So you can still see the Z disc. You can see that I band, the light band here, and then the A band, which is dark. You can see the M line. So the M line, remember M line, middle myosin. So right there. You can see Z disc to Z disc, so that is a sarcomere. So that's interesting. So let's take a look at the neuromuscular junction. So this is a synapse here. Specifically, it's a neuromuscular junction because this target cell, this is a muscle cell, and this is a neuron here. So it's a neuromuscular junction. Here is the synaptic cleft right here between these two. And also the axon terminal. So this is the axon terminal here, and these are synaptic vesicles. These are going to contain acetylcholine, which will be released into the synaptic cleft, go across here. It's going to bind to these ACH receptors. These ACH receptors can be actually disrupted with neuromuscular junction blockers. So some medications used for surgery actually act on these receptors so that you don't have a patient who is moving during important surgery or something like that. So it'll cause muscle paralysis actually. So a nerve signal is going to arrive at the axon terminal here. We're going to talk about the excitation of a muscle fiber. So we have a nerve signal arriving at the axon terminal, which is going to open up voltage-gated calcium channels. Calcium ions are going to rush in, and then the calcium is going to stimulate the synaptic vesicles. So these little yellow things here are going to actually release the neurotransmitter acetylcholine into the synapse here, into that synaptic cleft. The acetylcholine is going to travel, diffuse across, and it's actually going to bind to acetylcholine receptors on the other side. So acetylcholine, ACH, is going to diffuse across the synaptic cleft, and it's going to bind to these receptors here. So it's going to use 2 molecules of acetylcholine are going to bind to the receptor. Once it binds to the receptor, this is going to open this chemically gated ion channel, and this is going to create the end plate potential. So sodium's going to rush in, potassium's going to rush out, and that is going to that's going to create the end plate potential. Areas of the sarcolemma next to the end plate have voltage-gated ion channels that open in response to the end plate potential. Some of these are specific to sodium and emit it into the cell, while others are specific to potassium and allow it to leave. These ion movements create an action potential, as you can see here. The action potential is going to spread along the sarcolemma until it hits a T-tubule. Then it will be able to go down into the muscle cell here. It's going to hit that sarcoplasmic reticulum, those terminal cisternae, and it's going to, the action potential is going to open voltage-gated ion channels in the T-tubules that are linked to calcium channels. Then we're going to release calcium from those, from the terminal cisternae of the sarcoplasmic reticulum, and then it's going to go, the calcium's going to go into the sarcoplasm. Calcium is going to be in the sarcoplasm and the cytosol, and it's going to go throughout the muscle cell. Here it's going to bind, once it gets to troponin, it'll bind to troponin, which will cause the tropomyosin to move out of the way and expose the active sites. So here you can see the active sites are available for these myosin heads to come up and actually bind with actin. So the myosin head must have an ATP molecule bound to it to initiate contraction, and then it also has to break down the ATP into ADP and inorganic phosphate to cock it back to get it ready to go. So here it is ready to bind to that active site here, and then you can see down here we have the formation of the myosin-actin cross bridge. Now we're starting down here, so the myosin head is going to go through a power stroke, just like if you were grabbing a rope and you grab the rope, you pull it towards you, you release. If you were grabbing a rope with one arm, you would have to grab the rope, pull it towards you, release. That's a very similar action that this myosin head is doing. So this is the power stroke. It's going to get rid of, it's going to use up the ADP and P. It's flexing, it's doing the power stroke, and then it's still stuck to the actin binding site here. It needs to have ATP has to come up and hit the head, connect with the head, and then that'll knock it off. That'll break the bound myosin head. So now let's talk about how relaxation starts. So the nerve stimulation is going to stop, and therefore you're going to stop releasing the ACH. Those synaptic vesicles are going to stop releasing the ACH across that synaptic cleft. Also, the ACH that is in the synaptic cleft is going to start to be broken down by ACH, acetylcholine esterase. So that'll start the process of relaxation. We're also going to have reabsorption of calcium ions by that sarcoplasmic reticulum. And the sarcoplasmic reticulum is simultaneously releasing and reabsorbing calcium. But when that nerve fiber stops firing, calcium release also ceases, and only its reabsorption is going to continue. So we're going to stop releasing that calcium, we're just going to keep reabsorbing it. So since that sarcoplasmic reticulum is going to stop releasing the calcium, it's not going to bind with troponin, and if the calcium doesn't bind with troponin, then we're going to have tropomyosin is going to actually cover up those active binding sites, so then the myosin head can't come up and bind with actin. So that's going to further along the relaxation process. So now let's talk about the length-tension relationship and muscle tone. So the length-tension relationship, so the amount of tension generated by a muscle depends on how stretched or shortened it was before it was stimulated. For example, if overly shortened before stimulated, a weak contraction results as thick filaments just butt up against the Z discs. Another example, if two stretched before stimulated, a weak contraction results as minimal overlap between thick and thin filaments results in minimal cross-bridge formation. So the optimum resting length produces the greatest force when muscle contracts. And the nervous system is maintaining this muscle tone. It's maintaining that partial contraction to ensure that the resting muscles are near this optimal length. And here we can see the illustration of the length-tension relationship. We can see that optimal resting length right here. And we can see when it's overly contracted, it looks like this. And when it's overly stretched, it looks like that. And that makes sense. I mean, if you're overly stretched like this, you don't have as many heads coming up here to really pull these myofilaments together. And if you're already overly contracted, you're not gonna generate that much tension. Now let's get rid of the, let's zoom out a little bit and talk more of like a gross view here. So points of an attachment here. So origin is the less movable attachment point. So for example, the gastrocnemius is going to originate on the femur up here. You can't actually see it, but it's going to originate up there, and it's going to insert. The insertion is the more movable attachment. And it's usually distally, which it is in this case. So it's going to insert on the calcaneus here, the bone. It's known as the calcaneus right there. You guys already learned that for the last unit here. It's important to remember that skeletal muscles are attached to bones by tendons. So this is the Achilles tendon right here. Now let's talk about how muscles are named. So here, for example, our example is the sternocleidomastoid. It actually originates on the sternum and the clavicle. So sternocleido, sterno meaning sternum, cleido meaning clavicle, and then it's going to insert on the mastoid process right here. So this gives away what the name is, sternocleidomastoid. The origin and insertion give that away. We're also going to go over some other ways that we can figure out what muscles are named just by some of the clues that we can see visually. So other muscles are named off of the location or region. Like the pectoralis muscle is named because it's in the pectoralis region. The gluteus maximus, gluteus, the glutes, the gluteal region. The brachii, meaning arm, so the arm region. Femoris, we're talking about the femur here, so it's going to be near the femur. So these all give away hints to what the name of the muscle. Abdominis muscles, the abdominal region. We can also look at the actions. So if the action is a flexor or an extensor, adductor, abductor, those can be in the names, so those can help us figure that out. The fibers, the direction they're going, so rectus would be straight up and down, so rectus abdominis. Transverse, so we're going horizontally across, transverse abdominis. We have obliques, so going in a diagonal plane here. And then we have orbicularis oris, so orbicularis in a circular motion. We have orbicularis oris, we have orbicularis oculi, so those are two examples of that. And then we also have divisions as well. So we have the biceps, meaning bi, meaning 2, triceps, meaning 3, and quadriceps, meaning 4. So the size can also give us some clues as well. So vastus meaning huge, vastus lateralis, maximus meaning large, gluteus maximus, minimus meaning smaller, like the gluteus minimus, longus meaning long, such as the abductor pallicus longus, and then brevis meaning like brief or short. An example of that would be the abductor pallicus brevis. Shape can also offer some clues, like your deltoid is a triangular shape, so your deltoid muscle up here. Your rhomboid, your rhombus, a rhombus shape. Trapezius is in a trapezoid, so here's your trapezius right here.